Finally, it's time to talk about fatty acid metabolism. We'll start with its synthesis, so making fatty acids and triglycerides, and then we'll talk about its breakdown. When we discuss the making, that's going to be happening predominantly in your liver and your fat cells, and then your other tissues are going to be able to break down those. And so the making is going to happen in your cytoplasm, and as we'll see, it's also going to involve your mitochondria because of that pesky little deal with the acetyl-CoA not being able to enter or leave the mitochondria, but you make it in the mitochondria, but then you can't take it out of the mitochondria. So then we have the whole citrate complication. Don't worry, we'll get there, and hopefully it'll make more sense when we get there. But backing up, that was basically our synthesis of fatty acids. And if we want to talk about the actual breakdown of fatty acids, well, that's actually going to occur all in the mitochondria. And that happens in a process called beta oxidation. So we'll talk more about each of these, and we'll talk about the fatty acid breakdown in the next video. But today, let's focus on the synthesis. So again, we're going to be focusing on our liver and on our fat cells. The way that we synthesize these is going to seem a little bit weird, okay? So bear with me. So we've been talking over and over about how we build fatty acids based on acetyl-CoA units. And that's still true, but as we'll see, it's going to be a little more complicated because we're actually going to be building them from malonyl-CoAs. So let me explain. First, I have a few things to say about CoA. So CoA is going to show up a lot of different places in our metabolism. CoA itself is this bigger thing that's derived from vitamin B5 or pantothenate. It has this big kind of handle that you can use to have specific binding sites that will recognize this CoA and hold on to it. But really, the important part is just the sulfur right here. Why is it important? Well, sulfurs are swappable. Sulfur is right underneath oxygen on our periodic table. So same column, but different rows. Being below oxygen means that sulfur basically has an extra shell. Its electrons are going to be further out. So although it has the same number of valence electrons, those valence electrons are going to be more reactive. So sulfur is going to be more nucleophilic. It's also going to be, because it's bigger, it's going to be harder for sulfur to form effective covalent bonds. Remember that covalent bond formation involves the kind of overlap of electron clouds and orbitals. And well, when you have a sulfur trying to bond with something smaller, then you're kind of in a situation where you have someone with a really, really big hand trying to shake hands with a little kid. It's going to be hard. So those thio bonds to this sulfur are going to be weaker. They're going to be easier to break. And sulfur is going to often be the one breaking those bonds. So you can have sulfurs be swapping each other out, as well as have sulfur kind of make for a vulnerable point at which you can do things like form esters, form amides, and form carbon-carbon bonds. In the case that we saw before, we saw how pyruvate can get converted into acetyl-CoA, where we had an acetyl group put on CoA. Acetyl group is just one form of an acyl group that we can stick on CoA. So we'll see later how we'll have fatty acyl-CoA, where basically we stick a big fatty acid onto a CoA group. An acyl group, remember, is just where we have a carbonyl carbon attached to some hydrocarbon group. And then in the case of our CoAs, our acyl-CoAs, this is then going to be a CoA over here. So we saw how we could basically take pyruvate and we could transfer it to CoA and enter the tricarboxylic acid cycle that way. Let's also, though, today talk about what else we can do with acetyl-CoA. One of the other things that we can do with acetyl-CoA is actually use it to generate lipids. So we'll talk about lipid synthesis. We'll also talk about how lipids can be broken down to give you acetyl-CoA. And we'll talk about how some proteins can also be broken down to give you acetyl-CoA. So acetyl-CoA is really kind of central to a lot of different things in metabolism. And let's dive in. First, let's talk about where things are happening. Remember that when we talked about glycolysis, that was happening in the cytosol. And we talked about the citric acid cycle, that was happening in the mitochondrial matrix. In the case of fatty acid synthesis, the synthesis itself is going to occur in the cytosol, And it's going to happen predominantly in our liver and adipose tissues, so our fat cells. The breakdown of fatty acids happens through a process called beta oxidation, and that's going to be happening in the mitochondria. What happens is that your liver and your fat cells, they're able to synthesize these lipids, and they store them in the form of triacylglycerides. So remember, that's where we had 3 lipids on a glycerol backbone, and that was going to be very hydrophobic, allowed us to kind of like store these fats in these lipid droplets that aren't going to hog up water, be this very compact form of storage. When your cells have excess energy, then your liver and fat cells will build up more of this fat and store it. When other tissues need that energy, basically those fatty acids can be delivered. Instead of delivering just those globs of fat, they're typically delivered by chopping off the fatty acids, so by like lipases, and then transferring those fatty acid tails held onto proteins in the bloodstream, such as serum albumin. These fatty acids can then travel to the tissues can take them up, and they can do things with them. They can break them down, or the tissues can also incorporate them to make things that they need. Maybe they need membrane lipids, so our like phospholipids, incorporate them into more complex lipids, which occurs in the ER, or the endoplasmic reticulum. That's where we're going. Let's start, though, with just talking about this lipid synthesis. What do we need to do to make a lipid? Well, to make a fatty acid, we're going to need acetyl-CoA. We'll talk later about how this isn't as simple as just taking that acetyl-CoA out of the mitochondria, because acetyl-CoA itself can't get in and out of the mitochondria. So we're actually going to take citrate out of the mitochondria, which we can then convert to give us our acetyl-CoA, which we can use to make our fatty acids. But in order to make lipids, well, other lipids, if we want, say, a membrane lipid, we're going to need to attach it onto a glycerol backbone. We also need to attach it onto a glycerol backbone in order to make our triglycerides. So remember that we had the glycerol backbone, it had those 3 OH groups. In the case of our triglycerides, we're hooking up 3 of these fatty acid tails, 1 per OH. In the case of our phospholipids, we'd hook up 2, and then we'd have the phospholipid head. It turns out that even if you want one of those triglycerides, you start with having glycerol 3-phosphate, so you have 1 phosphate group there already. And this glycerol 3-phosphate is coming from dihydroxyacetone phosphate, or DCAP, which remember was coming from glycolysis. So this was basically, this component comes from glycolysis, this component comes from the TCA, and so fatty acids or lipid synthesis is kind of at the intersection of both of these, and we'll see how it can kind of be regulated by both of these pathways as well. Acetyl-CoA can also be used to make cholesterol, other steroids. We're not really going to talk about that, but know that there's this enzyme HMG-CoA, which is really important for this. And this is the target of statin drugs. But let's go back and let's talk about how we make these fatty acids. This fatty acid synthesis, it gets a little complex looking, and so don't worry about the chemical details. The important things to know are one is that this is happening in this complex called the fatty acid synthase. In our bodies, this is actually carried out by one big polyprotein that has all these different enzymatic sites on it. So there's going to be a bunch of chemical reactions happening, but all of these are happening on the same enzyme, the same protein. Whereas in like bacteria, it's going to be broken up into different proteins. What's going to be happening is that we're going to be synthesizing these fatty acids 2 carbons at a time. When we talk about 2 carbons, well, you're probably thinking acetyl-CoA's got 2 carbons, right? So let's just piece a bunch of acetyl-CoA's together. Somewhat confusingly, you're actually taking 1 acetyl-CoA, and instead of adding another acetyl-CoA directly to it, you're adding this thing called milanoil-CoA. So if we look at milanoil-CoA and we look at acetyl-CoA, we can see that milanoil-CoA has got this extra carboxylic acid group. So it has an extra carbon, so it's a 3-carbon compound, but we're only going to add 2 of the carbons. So we're going to use energy in order to add this carbon dioxide, which we're then going to remove. This might seem kind of silly, but what's happening is that what we're doing here is basically we're spending this ATP in order to activate this carboxylic acid that we add on. When we add it on, we add it in the strategic position where we have a beta keto acid. So we have this carboxylic acid group with a ketone that's going to be in the alpha beta position. As we discussed the citric acid cycle, decarboxylation of a beta keto acid is thermodynamically favorable. And so this is going to have the energy, provide the energy that we need in order to join these carbon groups together. The carboxylation is happening with this acetyl-CoA carboxylase, and this is going to give us our milanoil-CoA. So we would have acetyl-CoA and we'd have milanoil-CoA, but we're not actually going to join them as acetyl-CoA and milanoil-CoA. Instead, we're first going to link them onto the protein. First, let's orient ourselves to where things are happening. So remember, this is happening in the fatty acid synthase, and it looks a bit like this. It's got this key part that's going to be this acyl carrier protein that's going to be the point of attachment for our milanoil groups. And then it's going to have over here, it's going to have a cysteine residue, and it's basically going to transfer the milanoil groups from this ACP to that cysteine residue. And the cysteine residue, well, we're going to have our growing chain. Our growing chain is going to be held here, and it's just going to keep getting longer and longer and longer until finally we get that final chain. But the incoming groups are bound to this acyl carrier protein and then moved over. And so you're building kind of from the bottom up. And the newest added ones are actually going to be closest to the carboxylic acid group when we go and we cut this off. But going back to how it's attached, it's going to be attached through one of those sulfur linkages. So before we were talking about how we had coenzyme A, and it was really great because it had this big sulfur, and sulfurs are swappable. And we actually have our milanoil-CoA and we have our acetyl-CoA, and we want to transfer that part, that swappable part, onto a different sulfur. And so in the case of a cysteine, yeah, we just attach things to the sulfur and the cysteine. In the case of the acyl carrier protein, it's actually not that part of the protein itself, but rather what the protein is holding on to that plays a starring role here. Remember CoA, how it basically, all we really cared about was the sulfur group, but then it had this big part sticking off that was derived from our vitamin B5? Well, basically what we have here, in the case of our ACP, it's bound to this pantothenic acid group. And so basically it's almost identical to our CoA, except that instead of being bound to a phospho-AMP, it's bound to a serine. So it's bound to a serine in this ACP protein, covalently attached this group, which has this sulfur that's going to be swappable. And so basically we can take the group from our milanoil-CoA, or from initially our acetyl-CoA, and then esterify it to this SH group. So swap out what was attached to CoA now to this ACP. So it's basically a swap for a swap, and of the pretty much exact same thing, but they're just attached in different ways. So with coenzyme A, it was free floating, whereas here it's attached to the protein. And when it's attached, well, now it can get shifted, it can get pushed over. So you get it initially attached to the ACP, then transferred over to the cysteine residue. Now you just keep doing that over and over as you bring the new groups in, transferring them over. With each transfer over, you're going to lose a CO2. You're going to use the energy from that favorable decarboxylation in order to drive this process. When you're driving this process, well, now you're also going to have to be reducing things along the way, because remember, we want our fatty acid. We're trying to make a saturated fatty acid. It's going to be fully reduced. So as we add this, we're going to then have to go from a carbon dioxide to a fully saturated, so like an alkane. This is going to require 2 reduction steps. And to do this, we're going to use NADPH. So remember that NADPH was similar to NADH, except that it had this extra phosphate group. This phosphate group was allowing us ourselves to kind of distinguish between these two molecules. And this was important because, well, we wanted to use that NAD typically for our oxidative reactions, things like glycolysis in the citric acid cycle. In order to have enough of that NAD plus in that form, we would need to keep a high concentration of NAD plus compared to NADH. This was going to make it so that we could have those reactions be thermodynamically favorable. But in the case of our fatty acid synthesis, this is going to be a reductive reaction. So we would need to have NADH levels high. But we wouldn't want NADH levels to be high if we want to have this NAD plus ready to use to keep those other reactions favorable. So remember that glycolysis and this lipid synthesis are both happening in the cytoplasm. And so how are we going to rectify this? We're going to use NADPH instead of NADH. We can keep a high concentration of NADPH compared to NADP+, and therefore we have this pool that we're able to use. Basically, we just go through these reductive steps to get this saturated. And then we just keep doing this over and over and over. Well, not forever. Instead, this synthase is going to stop at this kind of like default point of palmitate. So the 16-carbon fatty acid, at which point palmitoyl thioesterase is going to hydrolyze it off, leaving us with this fatty acid, this palmitate. So to review, we have that setup phase where we have our fatty acid synthase is going to get attached to an acetyl-CoA. Well, at least it's going to get attached to the acetyl part. This is the only time at which we're actually just adding an acetyl part, like an acetyl-CoA, not a milanoil-CoA. So we add the acetyl part from acetyl-CoA, and this gives us our acetyl acyl carrier protein. At this point, we need to free up that acyl carrier protein site in order to add another, more carbons, basically. In this case, though, we're going to be adding the milanoil part of milanoil-CoA. So we've opened up that site by shifting things over to that cysteine residue. At this point, we have our acyl carrier protein site freed up, and now all we need is a milanoil-CoA. In order to get the milanoil-CoA, what we need to do is we need to activate an acetyl-CoA. That happens with our acetyl-CoA carboxylase, or ACC. And this is going to be an important site of regulation, as we'll discuss more in a minute. So how that activation occurs is it uses a bicarb, so basically your CO2. It takes ATP and it makes our milanoil-CoA. Seems silly, we're investing energy to make this milanoil-CoA because later we're actually going to release this carbon, so that carbon that got added from the CO2 is not actually going to get incorporated into the fat. Instead, what's going to happen is that the milanoil part of the milanoil-CoA is going to get linked up to this acyl carrier protein. Then what's going to happen is that the carbons that came from the acetyl-CoA, those are going to get attached to the carbons from the initial acetyl-CoA that was on that sulfur site, the cysteine site, and releasing that CO2. Releasing as CO2, the carbons that came from the milanoil part. So when we make this milanoil-CoA, basically what we're doing is we're setting ourselves up to have that energetically favorable linkage. If we look at the structure of milanoil-CoA, we see that we have a beta keto acid. That is, we have a ketone that is alpha beta to the carboxylic acid that we're going to lose. As we talked about when we talked about the citric acid cycle and when we talked about glycolysis, decarboxylation of a beta keto acid is energetically favorable due to the enol keto tautomerization that you get with that decarboxylation. Therefore, if we decarboxylate the milanoil-CoA or the milanoyl acyl carrier protein, basically that's going to then be energetically favorable. So this is energetically favorable, and we use the favorability of that in order to kind of link together these chains. How it happens is basically that the acetyl-CoA is going, or the acetyl acyl carrier protein is going to kind of attack the attached chain that was linked up to the other cysteine. And then because it's the one doing the attacking, now you're hooked up to the acyl carrier protein. Now what you need to do is you just need to kind of do some reductions. That's going to happen with our NADPH, which remember was one of the reasons why we need to do the pentose phosphate pathway was to regenerate NADPH. But this NADPH is now going to be used in order to reduce and dehydrate the ketone. So when we formed this bond, we formed a ketone. If we look at the structure of a fatty acid, you don't see ketones in there. Instead, if you had ketones in there, then the fatty acid would already be partly oxidized, and it wouldn't be as good of a fuel source. It also wouldn't be as kind of hydrophobic, so it wouldn't be as good for storing energy without storing water. So basically what we're going to do is we're going to use NADPH in order to reduce and dehydrate the ketone. When we do this, we now have this dehydrated. So we have this like saturated fatty acid that's kind of attached to, or I guess it's not a fatty acid at this point, but this saturated hydrocarbon that's attached to the acyl carrier protein of your fatty acid synthase. At this point, you need to free up that acyl carrier protein site, and so you just shift things over to that cysteine, just like you did before. Do this again and again and again and again. So each time you do it again, you basically have to use acetyl-CoA carboxylase to activate an acetyl-CoA to give you a milanoil-CoA. Attach that, link it up, reduce it, shift it, do it again and again and again. The default is kind of to stop at 16 carbons, which is the saturated fatty acid called palmitate. And at this point, thioesterase, which is part of the fatty acid synthase, is going to cut it off. If you wanted to add, say, saturation, so maybe you wanted to add a double bond, that would happen in your ER, and you could add those double bonds. Our cells actually can't introduce double bonds beyond the 9th or the 10th carbon position. When we talked about our fatty acid nomenclature, remember how we said there was like that omega nomenclature, which was where we were counting from kind of like the end, not the carboxylic acid end? Well, if we're counting from the end, and we said that we can't put past 9, put a double bond past between 9 and 10, well, this means that if you wanted, say, an omega-3 fatty acid or an omega-6 fatty acid, and you had a long fatty acid, well, then your body couldn't actually do that. And so that's why you would need to eat those omega-3 and those omega-6 fatty acids in order to have precursors. You could then modify those, but the omega-3 and the omega-6, those basically wouldn't be made. You couldn't make those from scratch in your cells because you wouldn't be able to add double bonds past the 1, 2, 3, 4, 5, 6, 7, 8, 9, past like right here. And so if you wanted a double bond here, you would have to actually eat a fatty acid that had a double bond there. Because although we can elongate fatty acids, that elongation, just like when we were making it, that elongation is going to happen from the end that's attached to the carboxylic acid. Remember that this is where the carboxylic acid is going to be. This is the end that your elongase would add to. And you can't add the double bonds over here at the very end over here. And so you would have to eat those fatty acids in order to get those. And so that's why we consider those like dietarily essential. We needed acetyl-CoA to start that all, but acetyl-CoA can't enter or leave the mitochondria. We talked about this when we discussed how when we did the oxidative decarboxylation of pyruvate, we did it inside of the mitochondrial matrix because that CoA couldn't get through. So instead of trying to transport this acetyl-CoA through, we're going to transfer something we can convert into acetyl-CoA. So remember, we're generating this acetyl-CoA in the mitochondrial matrix, and now we want to get it out. How we could get it out is in the form of this citrate. So remember that in the tricarboxylic acid cycle, we combine acetyl-CoA, we combine the acetyl group with oxaloacetate to give a citrate. We can take this citrate out of the mitochondrial matrix, and then we can use that citrate to basically reverse things to separate it back into oxaloacetate and acetyl-CoA, and then we could use the acetyl-CoA. When we're doing this, we can do it using this enzyme ATP citrate lyase, which will produce our acetyl-CoA and our oxaloacetate. As we discussed when we're talking about the TCA cycle, oxaloacetate can't get in and out either. It's going to be highly regulated. And so instead, we're going to need to, if we want to get that, the rest of the carbons back into the matrix where they can be broken down further, oxidized further, we're going to need to transport them in a different form. We have kind of two options. We can convert it to malate or we can convert it to pyruvate. Either way, we're actually going to convert it into malate first. This is the same reaction we were looking at when we were talking about how we get NADH equivalents into the mitochondrial matrix via the malate aspartate shuttle. Remember there, we took oxaloacetate and we took NADH, and we reduced oxaloacetate to malate. Malate was then able to sneak those electrons into the mitochondrial matrix, where they could be transferred back onto NAD plus to give us NADH. The reason why we were doing it there was basically to regenerate that NADH inside of the mitochondrial matrix. That's not really our goal here. If you have malate and you send it through, you're going to get more energy than if you send pyruvate through, because malate's got more carbons in it. But what if what you care about is not so much those carbons, that energy? You've got plenty of that energy if you're making fats, probably. Instead, you're going to want that NADPH. And so what you want to do is basically take this malate and you're going to oxidize it. But you're going to oxidize it using NADP plus to give you NADPH. This takes place with this enzyme, malic enzyme. You're getting this NADPH, which you can use to power the lipid synthesis. That's not the only way we can get NADPH. Another key way is going to be the pentose phosphate pathway. Putting this into the bigger context, all of this is kind of happening surrounding acetyl-CoA. But that acetyl-CoA, remember, can be coming from pyruvate if we wanted to, say, make fats from our sugars. It could also be coming from some amino acids. If we want, however, to make fats from sugar, well, we make pyruvate. And we make pyruvate, we transform pyruvate into acetyl-CoA through our pyruvate dehydrogenase step. But that was happening inside of our mitochondria, because we couldn't do that outside of the mitochondria, because we couldn't basically take acetyl-CoA in or out of the mitochondria. Now, this is great if you actually want to do something in your mitochondria, like you want to do your citric acid cycle. It's great that you already have your acetyl-CoA here. But when we talk about building fatty acids, that's actually happening in our cytosol. And so if we want to build a fatty acid from this acetyl-CoA, we're actually going to have to transport it out of the mitochondria. And then in the cytoplasm, we're going to basically hook it up. But we can't take acetyl-CoA out of the mitochondria. So instead, we have to basically hook that acetyl-CoA up with oxaloacetate to get citrate, and take the citrate out of the mitochondria. Once we're out of the mitochondria, well, now we can convert it to acetyl-CoA, and now we can use that acetyl-CoA in order to make our fatty acids. So we kind of have to go this roundabout route where we take that citrate out, and then we use ATP citrate lyase in order to make oxaloacetate. As I mentioned, this acetyl-CoA carboxylase, or this ACC, is going to be an important site of regulation, and it's also going to make an important regulatory molecule that's going to regulate our beta oxidation or our breakdown. Basically, we're going to get reciprocal regulation, such like we saw a bunch of reciprocal regulation when we talked about like glycogen metabolism. We're going to get reciprocal regulation in terms of our beta oxidation, so our fatty acid breakdown, and our lipogenesis, so kind of like making our lipids. And this is going to be largely carried out by this acetyl-CoA carboxylase complex, so this ACC, as well as by this CPT1 or CAT1, this carnitine acyltransferase, also known as carnitine palmitoyltransferase. We'll talk more about this in the next video, but basically this is going to be how you import fatty acids into your mitochondria in order to oxidize them. And we'll see that the milanoyl-CoA that we formed during the process of making amino acids is going to inhibit that process, so that we inhibit the breakdown when we're making these fatty acids. For now, let's just focus on this ACC, this acetyl-CoA carboxylase. If we look at the diagram, we can see that we have examples of both feedback inhibition and feed-forward stimulation. The feedback inhibition, basically, if we're building up a lot of long fatty acids, basically that's going to inhibit our acetyl-CoA. And if we're building up a lot of citrate, that's going to be feed-forward stimulation. We've got a lot of citrate coming. Go ahead and get ready for us. So activate that ACC. These are both examples of allosteric regulation. However, ACC is also going to be regulated covalently in response to hormones. That acetyl-CoA carboxylase, it can basically be in a monomer form, which is low activity, or it can be polymerized, which is going to be a high activity. When it's phosphorylated, it tends to be more of like the monomer form and more inactive. When it gets dephosphorylated, well, now it gets activated, it gets it polymerizes, and you basically get the melanoid-CoA formation. When the melanoid-CoA is formed, it can stimulate it be used to make these fatty acids, and it can also go and it can inhibit the breakdown of fatty acids. If we look about what this is going to be modified by, well, in this case, we are going to want to think about what hormones we would want to stimulate this. If we talk about building fatty acids, when would we want to do that? We would want to do that when we have plenty of fuel. We would want to do that when, say, the blood sugar levels were high. We wouldn't want to do that when the blood sugar levels are low. In that case, we wouldn't be breaking down fatty acids. And we wouldn't want to be doing that under like a stressful situation where we need to run away from a bear, in which case we would also want to be breaking down fatty acids to get some energy. So we want to find a way in which we could stimulate this process in response to insulin, which remember is released in response to kind of like high energy. And then we want to inhibit it in response to glucagon, or like low energy, low blood sugar status symbol, or epinephrine, so also aka adrenaline, running from that bear. And how we can do this is through phosphorylation, regulation of the kinases and phosphatases. So we said that the phosphorylated form was inactive and the dephosphorylated form was active. This might remind you of some of the enzymes when we talked about glycogenesis and glycogenolysis. How, for example, we had that glycogen synthase was going to be inactive when it was phosphorylated and activated when it was dephosphorylated. In that case, we saw that pKa, which was activated in response to adrenaline or glucagon, that was going to phosphorylate and inactivate glycogen synthase, so we didn't make glycogen. Conversely, insulin was going to activate phosphoprotein phosphatase, which was going to dephosphorylate glycogen synthase, activate it, so we can make glycogen. We see something very similar happening here. In response to the epinephrine or the glucagon, you're going to activate pKa. pKa can now phosphorylate your acetyl-CoA carboxylase, and in response to this acetyl-CoA carboxylase phosphorylation, now you're going to inactivate the acetyl-CoA carboxylase. So if you have low energy, if you need to make energy, you want to be breaking down fatty acids, you don't want to be building them up. And if we want to break down fatty acids, not only would it be counterproductive to build them up, but the melanoid-CoA that we form would actually inhibit the breakdown. So we need to make sure that we shut that down, and we can do that by phosphorylating the acetyl-CoA carboxylase. However, we need to be able to reverse the signal. We can reverse the signal by having insulin activate a phosphatase. The phosphatase is going to dephosphorylate the acetyl-CoA carboxylase. This is going to activate it, and in response to that activation, now you can go and you can build the fatty acids. So that was an example of covalent modification and hormonal regulation. On the other hand, we also saw allosteric modification in the case of our citrate, where we had the feed-forward stimulation, and in the case of our feedback inhibition with our palmitoyl, so our long fatty acid feedback inhibition. That palmitoyl-CoA was going to favor that low activity monomer, whereas the citrate was going to favor the high activity polymer. And then the phosphorylation was also going to kind of influence the activity of this enzyme. And that way you're able to integrate signals from all sorts of different things, whether it be your hormones or whether it be the concentration of the various components in the cell at that moment. So that is going to be the key regulatory point of our fatty acid synthesis, is going to be this step where we have this acetyl-CoA carboxylase. And next lesson, we'll talk about how the key step of regulation for our beta oxidation, our fatty acid breakdown, is going to be the CPT1, also called CAT1, your carnitine acyltransferase, which is going to bring the fatty acids into the mitochondria in order to be broken down. It's going to be regulated by melanoid-CoA, and negatively regulated by it. So basically, when we're building fatty acids, we don't want to inhibit the breakdown. We want to inhibit the breakdown of the fatty acids. But if we build up too much fatty acids, then we want to inhibit making more metabolism. It's cool, right?